Hello everybody, it's Luke D, and this is Honors Bio 2 Mechanisms of Cancer. It's 2012, November 11th, and we are going to be talking about epigenetics today and its relationship to the cell cycle. We got kind of snagged up and did not get to talk a lot about some of the experiments that we had first talked about. I wanted to leave you kind of in a questioning state and then bring you back, but unfortunately I just kind of left you in that questioning state. So let's look at this cell cycle really quickly. You can go around the cell cycle, clearly starting with a mitogen signal, starting with G1, S, G2, mitosis, and then cytokinesis. You guys know that there's exit from the cell cycle, and what that means is that the cell arrests somewhere here. But certainly before synthesis. Now that's really important. A lot of cells, probably most of our cells in the body, have actually arrested, which is good because let's say you have a brain cell, you don't want it to rapidly divide because there's only so much room in your skull. If a cell rapidly divides, then you no longer have space in your skull and you die. We also know that there are other exits from the cell cycle. We know that there are checkpoints, things that can stop it, and we know that there are ways to, once you've stopped the cell cycle, to commit the cell to apoptosis, which again is cell suicide. Remember, we likened that to being in a room, in a school, and having one bad kid in a room and then blowing up that room to destroy that bad kid. And in this case of the human, that's very much worth it. And it might be even worth it to do larger preventative maintenance because clearly cancer is a problem. So, how does that have to do anything with epigenetics? Well, let's look. Alright, so we looked at this really cool experiment, and this is where we took the two moms and the two dads and we saw if we could have babies with them, which would be awesome for a lot of people, but not so great for the embryo. Because as you can see here, the two moms don't generate a placenta, or a good placenta, and the two dads don't generate a good embryo. I kind of think this one looks like a pirate, so this is the head, and this is the eye patch, and that's the beard, mouth, and then an eye would go right there. Anyway, back to what we were talking about. The reason that this could generate, this two-daddy one could become like this, is if there were 22 chromosomes and a Y on both genomes. But that's not the case. These are 22 chromosomes and an X. So this is a complete genome. I guess you could call it a complete, yeah, we'll just call it a complete genome. Now, if I told you that these were actually basically clones of each other, except for the sex parts, it wouldn't make sense for this to generate cleanly and this embryo to just not be put together at all. You could expect that it was because of gene expression, so different signals here, or different cocktails of signals here, resulted in this, and then a different set of cocktails resulted in this, but that's not the case. These are experiencing the same maternal environment. So we know that the signals it's receiving are the same, which means that it's not the interaction between the environment and the DNA producing certain genes. That means it's got to be something else, and you guys hit upon this a long time ago. But basically, you said that the maternal genes had the placental genes shut down permanently, and that the father had some of the embryonic developmental genes shut down permanently. So let's keep that in mind. Big takeaway here, mom genome has some parts of it that are shut down, that are specific to moms. Dad genome has the same. Next experiment we looked at was this Blastomere isolation experiment. So we took the cells, we separated them by shaking them hard, and then one of them dies. The other one becomes a Blastomere and then a sea urchin. I believe that's called a medusa. I can't remember though. But how does that happen? How does one cell always die? Because these look identical. They're clearly not, because one is always dying. So how is that happening? And we know that their nuclear contents are exactly the same. So it can't be the nucleus that's different. But what we ended up figuring out was that the cytoplasm of these two were different. So let's say this was one cell in the beginning, and let's say it produced a bunch of A protein from this side and a bunch of B protein from this side. Well, then when they split, this one would have a lot of protein A and this one would have a lot of protein B. For some reason, protein B maybe shuts down the genome so that this thing can't express what it needs to express and that it dies. So, we know that this epigenetic mechanism can shut down parts of the genome. We also know that the cytoplasm helps to determine parts of the epigenetic mechanism. So we know that things in the cytoplasm can affect the DNA in such a way that some cells will live and some cells will die. This further goes on, this is further clarified in this experiment. So if you take a skin cell and you put it on a petri dish and you culture it, it'll stay a skin cell. It might stay there for a long time, but it'll still be a skin cell. So what you do is you take an egg, you zap the nucleus with UV radiation, which just shreds DNA, which is why you should wear sunscreen, especially if you're pale. And I forgot where I was. Anyway, you take a nucleus out of that, you take an egg with a destroyed nucleus, and you put the nucleus from here into this egg, and guess what? You get a whole tadpole. So we know that this cell, that is a skin cell, or that was a skin cell, must have moved far beyond this point. Far beyond this point. So it would no longer, so it has a good deal of its genome shut down epigenetically. But somehow, being in the egg juice in here allows it to unshut down. How that mechanism happens, we'll talk about later. Moving to here, we can see that certain signals from certain parts of the cell will actually force the cells around it to develop in a certain way. So, for example, this is the nucleus center, which is going to dorsalize or make like the back the area here. Okay, so you got one nucleus center, which means you're going to end up with one dorsal side. So, one nucleus center, one dorsal side. And what happens if you transplant a nucleus from another cell into there, or from another organism there? Well, guess what? You get nucleus, you get dorsalization on two sides. Two nucleus centers, two dorsal sides. And again, what that means is that this would be like the back or spine of an organism, and this one essentially has two spines connected down here at the bottom. Alright, what does that mean? Well, if we're saying that skin cells have somehow epigenetically had their genome shut down, and we know that they can be unshut down by the juice of an unfertilized egg, well, what we're seeing here is that these cells can be forced into epigenetic shutdown, and thus be a certain kind of cells by some of the information that other cells around it are secreting. I hope that was clear. Basically, you can make egg juice unshut down a genome, and then you can have other signals which will help to shut down a genome. These egg juice factors are located in the egg cytoplasm. These are secreted from the cell. We have the same kind of idea here. So how does that relate to our cell cycle? Well, a cell that has epigenetically shut down some of its ability to replicate will proceed in G0 unless those epigenetic mechanisms are somehow restored. And if they're restored in full, the cell will become completely functional, but will still depend on a mitogen or start signal in order to... Oh, sorry, I was trying to get rid of the start Okay. So it would still depend on a mitogen or start signal to start. And if you think about it, a stem cell is a lot like cancer. So if you take a stem cell, you can make any kind of tissue out of it. You can make a head, you can make an eye, you can make liver, or whatever you want. And cancers have theoretically the same ability. For example, there's one called a teratoma. If you look it up, I'm going to trust you to be adult, but it's quite gross. There's a tumor that can produce different types of tissue in the wrong place. So you might have teeth growing on the inside of your intestine, for example. If you've been to the Bodies exhibit, there's actually a display of it. So a stem cell that generates you divides, unevenly splits its cytoplasm, and then in a series of really highly organized events continues to divide as the cells, some become differentiated and become head, hair, other ones, as the organism gets bigger. Some will remain stem cells, as we saw in the case of the intestine. We need to be able to regenerate those intestine cells, aren't they? Those down, yeah. So here, remember, here is the stem cells. They are producing all these cells that are actually changing depending on the amount of signals they receive as they go farther away from beta-catenin and TCF. Okay. A cancer cell, however, if you put in the womb, will not generate a child. Why? Well, a cancer cell has the ability to replicate, but it does not have appropriate things shut down, and given the right signals, it will still not produce a human being. So, the relationship between these two sets of things, the cell cycle, and these experiments, is that they highlight some of the differences between limited growth, which is how you come from an egg, and unlimited, undifferentiated growth, which is how a tumor comes out of a cell anywhere in your body. We know that there's got to be epigenetic mechanisms at play. However, we don't know right now what all of them are. Okay, I hope that was all clear. If not, hit rewind and give it a shot again. I am going to shut this down. I'll see you later.